good evening to everyone who are here in this live session. I hope the audio is clear. Can I get some thumbs up so that I confirm that the audio is clear? Thank you. Once again, formally welcome you all to this live session for the Applied Environmental Microbiology course. And I am Guru Priya, a Prime Minister's Research Fellow in the Department of Civil Engineering, IIT Madras. I have my BTech in Biotechnology from Kalasalingam Academy of Research and Education, Tamil Nadu, and my MTech in Biotechnology from Indian Institute of Technology, Guwahati. So currently, my doctoral research focuses on enhancing biomethane production from organic wastes through anaerobic digestion. So this is a brief introduction about me, and in this course I have a module where it is more interactive, and I suppose that all of you would interact better so that all of us learn more and get to know things uh, in a clearer way. Before starting today's session with all these questions and discussions, let us have a brief introduction about you, your background, so that that will help me to prepare the future sessions accordingly. So the first session I have basically prepared based on my understanding. So if I know your background better, then maybe the questions that I frame and the concepts that I come with can be more aligned and be customized. So you can type your introduction, your name, I mean the course that you are currently enrolled in, or the job that you are doing. Uh, these informations basically through the chat box, or if you are comfortable, you can also turn on your mic and answer. Nice. Hi, Ansh. And anyone, if you have any vision or expectation from the session, you can also post that in the chat box so that I take note of that as well. So for those who have joined recently, was just asking for a little introduction about yourself, your name, and uh, the current course that you're enrolled in, or the job that you're doing, in the chat box. And I'm just waiting for the response. I suppose we are 12, and including me, we have some 5 responses. So I'd be happy if the rest of you could also post it in the chat box. Yes, Tulasi, you wanna say something? You can unmute yourself or post in the chat box. the remaining, if you still wanna share your introduction in the chat box, you can still do that. Now we'll formally start with the session. So before that, I'll just share my screen. Okay, so I guess now the screen is visible. Yeah, so this was my first slide to welcome you all. So, and it's really good to know that many of you are from life sciences, microbiology, or biotechnology background. I had handled the same course TA-ship in 2024 as well, where I had participants from even more diverse backgrounds. So based on that understanding, today's session could be a little easier for you because it's all just basics that we'll be discussing here. So the basic format of the sessions that I handle would be that I would bring up some questions and give you a couple of minutes, a few minutes to answer that question, for which you will be posting the answers in the chat box. And after we receive some significant amount of responses, either same or different, we'll then go about discussing the answers for that question. And I also have some divisions, like a subdivisions. So after each few questions, we'll be discussing one concept. It is not strictly that way, so you can, whenever you have a doubt, you can post in the chat box, or you can unmute yourself and ask. Or if already you have some questions as well, as soon as you come or whenever in the sessions, you can feel free to ask your doubts. So with that, I hope all of you went through the course content for week 1. So the course content basically talks about the history of environmental microbiology, microscopic techniques, about the cell structure and composition, about how cytoplasmic membranes are there and how they facilitate transport, followed by the important factors called DNA, genes, and genetic elements. And then finally, there was some discussion about the central dogma, which is the replication, transcription, and translation. So this is the overall course content that was discussed in the week 1 lecture in the Applied Environmental Microbiology course. So as said, I'll post some questions. You can just post the options in the chat box. So the first question is, identify the true statement regarding eukaryotes. you are telling it is C, true that DNA is bound with histones in eukaryotes. So I'm sure that you are aware of the differences between prokaryotes and eukaryotes. So we'll also see that in one of the summary slides. So basically, a prokaryotic organism is considered to be primitive because it lacks membrane-bound organelles like nucleus, mitochondria, and other membrane-bound organelles. And again, DNA is present in the eukaryotes. So in order to be compacted, there is a protein called histone around which the DNA is wound. And histone is a positively charged protein which allows the compaction of this DNA so that it can fit in the nucleus. So yes, C is the right answer. Okay, now you have to identify which of the following is or are prokaryotic organisms.
A and C is the answer.
Right? So A and C both are the prokaryote. I mean, yeah, one is the subset of other. Bacteria consists of gram positive and gram negative, whereas fungi can be unicellular, multicellular, but it's a eukaryote, and protozoans are unicellular organisms, but still they are eukaryotes. So fungi and protozoans are eukaryotic, whereas bacteria is prokaryotic. So the next question is which of the organelles has its own genetic material?
Mitochondria.
Exactly. as you mentioned, mitochondria is one of the organelles which has its own genetic material apart from the organism's genetic material present in the nucleus. So here is a summary of what is a prokaryote and a eukaryote. So typically the prokaryotic organisms have a cell size of 0.2 to 2 micrometer in diameter, whereas eukaryotic cells are bigger in size, ranging from 10 to 100 micrometer in diameter. Uh, so the stark difference would be that prokaryotic organisms have no nuclear membrane or nucleoli, and we just have the genetic material laid over in the cell, whereas the genetic material in eukaryotes are going to be present inside a true nucleus. And as we saw, prokaryotic organisms lack membrane bound organelles, whereas in eukaryotes we have different membrane bound organelles that perform various different functions. So these organelles include Golgi complex, mitochondria, endoplasmic reticulum, and chloroplasts. And also there is a difference in the flagella between prokaryotes and eukaryotes. So in prokaryotes it's very simple and consists of two protein building blocks, whereas eukaryotic flagella is a complex structure. And again, we have this glycocalyx, which is present as a capsule or a slime layer in prokaryotes, um, whereas in eukaryotes it's only present in some cells where they lack a cell wall. And similarly, cell wall also is usually present in prokaryotes, whereas it's not very commonly present in eukaryotic organisms. And the cell membrane or the plasma membrane has no carbohydrates and lacks sterols in the prokaryotes, whereas both of these serves as receptors in the eukaryotic cells. There is no cytoskeleton in the prokaryotes, and the ribosomes are going to be 70s ribosomes. And we have a single circular chromosome without histones in the prokaryotes, whereas we have multiple linear chromosomes with histones present in the eukaryotes. And the cell division process is going to occur via binary fission in prokaryotic organisms, whereas it involves mitosis in eukaryotes. And the sexual recombination is not happening in the prokaryotes, whereas it involves meiosis in eukaryotic organisms. So this is the basic difference that we have to know right before entering into environmental microbiology. I hope this is clear. So next, as we saw in one of the uh, questions, I just have a representation of fungal cell, protozoan cell, and a prokaryotic cell. So you can see that the prokaryotic cell has no membrane bound organelles. It's pretty freely seen in the um, image, whereas if you see compare the fungal and protozoan cells, you can see that there are different small organelles present inside the cell. So you can just compare this table and the image present here and understand the increase in complexity in the cell structure in the eukaryotes as compared to the prokaryotes. Any doubts so far?
No, ma'am.
No, ma'am.
Okay, so the next question is about three domain classification. Who described the three domain classification?
Robert Hooke, ma'am.
Okay, so uh, yeah, it's okay to answer in the answer through the mic, but it would be better if you could post in the chat box. as well. Like after answering in the mic, you can also post in the chat box so we can see if there are any varied answers. Your answer was C, and I can see a B in the chat box. Okay, we have couple of answers now, and mostly it is B. So yes, the three domain classification was brought about by Carl Woos. And uh, do you know what are the three domains that he classified organisms into? Bacteria, archaea, and yes, one more. Exactly, eukaryota. So Carl Woos was an American microbiologist, and he thought that it is important to classify organisms. Like this domain classification comes one step ahead of the uh, ahead in the taxonomic classifications before the kingdom level. So we usually start with kingdom, phylum, class, order, etc. So Carl Woos and his colleagues felt that there should be one more classification called the domain based classification even before the kingdom classification, and they proposed three domains which were archaea, bacteria, and eukarya. So archaea are again, archaea and bacteria both belong to prokaryotic, and eukaryotes are, yeah, obviously eukaryotic organisms. So archaea is single cell prokaryotes, and it is mostly found in extreme environments, whereas bacteria are traditionally single celled prokaryotes that we see around. We have this habit of calling all the microorganisms are bacteria, so that is not true. Microorganisms are microorganisms, in that genetically we can call some of them as archaea, some of them as bacteria, and maybe some unicellular organisms like protozoans, as we saw, can be eukaryotic also. And here in this three domain classification, the basis of classification was the 16s rRNA, which is the 16s ribosomal RNA. So based on the differences and similarities found in the 16s rRNA, they have made this classification, and even now we use this 16s rRNA sequencing, uh, rRNA based amplicon sequencing, uh, to find the microbial communities, or to find the microorganisms present in a random environmental sample. So that is about the three domain classification brought about by Carl Woos. So the next question is, the organisms that can survive in very low temperature conditions are called? Okay, let's see the answer. Um, okay, before seeing the answer, what do you have any idea of what are barophiles, extremophiles, and psychrophiles? You can just type in a chat box, like you just type baro and what it means, extremo, some short forms, psychro, whatever, and then type what they are.
Barophiles can thrive in a high pressure condition. Extremophile are those organisms that can thrive in extreme condition like extreme temperature, extreme pressure. Overall, they are living in a extremely difficult atmospheric condition.
Okay,
Psychrophiles are the cold-loving microorganisms.
okay, perfect. So we also have in chat box that extremophiles are organisms which can survive in high or low temperature. Okay, so suppose that pretty much all of you have the same opinion that barophiles can survive in extreme pressure conditions, psychrophiles thrive in extremely low temperatures, which was the question again. And extremophiles is kind of a umbrella term, right? Extremophiles, so barophiles can be called extremophiles, psychrophiles can be called extremophiles, thermophiles can be called extremophiles, acidophiles, alkaliphiles, whatever, they can be called extremophiles. So do you still think C is the right answer for this question?
It should be D. It should be actually all of the above, but in this case, D.
Yeah, all of the above, because you indirectly link temperature to some pressure or whatever, but leaving all those
Basically,
complications.
the question is the organism who can survive in a low temperature. So specifically, psychrophiles can thrive in a low temperature. The right answer is C.
No, it cannot only be C, because extremophiles is an umbrella term where all
Okay, okay, okay,
extreme organisms come under extremophiles. So in that case,
okay,
your answer should be D, where it's both B and C.
okay, okay. Thank you.
Yeah. Okay, I hope this is clear. If you still have any doubts, you can ask or you can post in the chat box. We can discuss. So for now, the answer would be both. The organism is an extremophile, which can specifically be termed as a psychrophile. The next question would be, MCRA gene is linked for methane production in methanogens. True or false? Okay, so this was purely a memory based question, and many of you may or may not know the answer. It is true that MCRA gene is linked with methane production in methanogens, and you will come across this particular gene example in quite a few places in the lectures, and that is why I wanted to reiterate this question here so that you can relate it and have it in your mind whenever you come across that. It is one of the most commonly cited examples in any environmental microbiology related topic. So, to give a few information about MCRA gene, it is a functional marker we use to study methanogens with, and basically this gene encodes for a alpha subunit of methyl coenzyme M reductase. So this methyl coenzyme M reductase is one of the key enzymes in uh, methane production. Uh, it's present in the archaea, and this MCRA gene codes for alpha subunit of that enzyme, and therefore it is a very critical functional marker uh, when characterizing methanogens or when studying methanogens and methane production. So with that, we come to the summary of this topic, where we saw about this three domain classification, which is archaea, bacteria, and eukarya. So we know the cell type of archaea and bacteria is prokaryotic, and the difference between archaea and bacteria lies in their cell wall, where bacteria has a peptidoglycan cell wall, whereas in archaea it's going to be varied in composition and there's going to be no peptidoglycan, and that makes all the difference. And the membrane lipids are branched carbon chains, and they are attached to glycerol by ether linkage in archaea, whereas in bacteria the chain is going to be straight, and it's going to be attached to the glycerol by ester linkage. So it's ether in archaea and ester linkage in bacteria. And one more difference is that the chain is going to be branched in archaea, whereas it's going to be straight in bacteria. And you can see one more similarity in terms of protein synthesis between archaea and eukarya, where methionine is going to be the first amino acid in protein synthesis. So this table gives the basic understanding of archaea, bacteria, and eukarya, and as we already saw, you can see in the top right the three kingdom classification and some examples in each of it. If there are any doubts, please post in the chat box. If not, we'll next go to the microscopy topic, where you can answer what is the range of an electron microscope. ended only. You can answer in terms of magnification or resolution. So we already have one answer. I'll wait for a couple of more answers. Okay, so we just have one answer for this question so far. So we'll see the answer. Before seeing the answer, we'll see some basics of microscopy. So we all know that a microscope is a device that we use to visualize microorganisms, microparticles, nanoparticles, all of these. So in our case, we are more concerned about microorganisms. We use different types of microscopy to visualize microorganisms and including their organelles, which can be in even lower ranges as well. So there are a few concepts that we have to know before dealing with microscopes. So first we should know what magnification the microscope works, and I'm pretty sure that all of you know what a magnification means. It means how big can we see the smallest particles that is under examination. So basically how enlarged the image can look like. It's the process. Magnification is basically the process of enlarging the apparent size of the object in the image. And we have different lenses in a microscope, one of which is the objective lens, and in total magnification we calculate the total magnification uh, by multiplying the magnifying power of the objective lens and the ocular lens. So what is an objective lens and the ocular lens? No answers? Okay, right. So ocular lens is near to the eye, or the lens through which you see into the image is the ocular lens, and the lens that is focused to the object is known as the objective lens. Okay, the object under examination is facing a lens called the objective lens. It's usually present in the bottom in a normal compound microscope, and in the top you have a lens through which you see into the image, and that is known as the ocular lens. Okay, so when you m- multiply the magnifying power here of both these, then you get the total magnification. That times magnified will your image be in comparison to the original object under the examination. So magnification, magnifying power is one important concept in microscopy, and the next one is the resolution or resolving power. So resolution is the ability to distinguish between two closely spaced points, which means I have a point uh, A. Let's say this near 7 there is a dot here, right? That is one point, and there is, imagine there is one more point below it. So if I can distinguish between 2 points uh, in a distinct way, then that is going to be the distance is going to be the resolving power. The minimum distance between that is going to be the resolving power. If both the points are going to look together, then the resolving power is not that high to distinguish both the points. So the second concept is about resolving power. Okay, you can answer the range of any microscope in either of these terms. So the answer for this question would be that the range of electron microscope is going to be 100 micrometers to 1 nanometer. I hope the basics was clear. If there are any doubts, please let me know. And the next question is the match of following question. So you can just match and give the right option 1, 2, 3, or 4 in the chat box. You basically have to match between the type of microscopy and some related concept based on which it works. here the microscopy has to be matched with the operation principle or the kind of image that it is going to give. So does anyone have any idea of what is a differential interference contrast microscopy and for what we use it for? Okay, so let me answer that. A differential interference contrast microscopy basically uses a polarized light, and it also uses some specialized prisms called Nomarsky or Wollaston prisms, where the light that you are trying to uh, throw on the sample will be split into different beams, and these beams will pass through adjacent parts of the specimen, and again, while coming to the image formation part, they recombine and interfere. So you send a light source and you split it into different beams which falls on the object, and again the split beams while turning as image are going to be recombined and they interfere. So this is basically differential interference contrast. So you know now that it can be attributed to refractive index because you're going to use a prism, you're going to polarize the light and split it into different beams. So there comes the concept of using the refractive index. Okay, and in case of atomic force microscopy, if you have already known, then you know that it's one of a very different kind of microscopy where it's not just imaging, it's just like something like touching and feeling. Though there could or could not be contact, there are different types of atomic force microscopy. But let's say you have a table, okay, and if it is a new table, it's going to be very smooth, but when you see some old wooden tables and all, you can see some parts of it being broken or taken away, some small parts of it. So that gives kind of a rough uneven surface to the table, right? So when you have a pen or a brush whatever in your hand and you're going to go through the surface of the table, you're going to constantly face some kind of ups and downs while traversing through the surface of the table. So similarly, when you're going to have a specimen under examination, the surface of it need not be very smooth and even, right? So it can be uh, with some crest troughs, some ridges or whatever. In atomic force microscopy, you will have a sharp probe with a tip. So this is going to be attached to a cantilever, and it's going to straight constantly move through the surface of the specimen, and there will be a laser light that is focused on the cantilever. So when you're going to move through it, if there is some depression, your probe will sense that depression and the cantilever will move down. Again, when there is some kind of a bump, it's again going to move up, and the laser that is focused on this cantilever is going to sense these differences based on the movement of the cantilever in up and down direction. Okay, and this laser beam that is being read in the detector source will be used to construct the topographic image. Okay, so in that case, the atomic force microscopy is for topographic mapping. And in case of confocal scanning laser microscopy, you will have laser scans through the sample in each point, and in addition to that, you will have a pinhole aperture, and this pinhole aperture will block the out of focus light. So why are we blocking the out of focus light? Just to ensure that the light from a thin optical section is only detected. So you can see stark differences using confocal scanning laser microscopy, and for this you will also need the sample to be colored. Okay, so there the sample contains some fluorophores, and for C it's going to be 4. And finally, for electron microscope, it's very common that we have two different types of electron microscopy: scanning and transmission. So in electron microscopy, the source is not any form of light, it's going to be electrons. So if the electron is going to fall on the surface and then come back to the detector in the opposite direction, you call it as a scanning electron microscopy. It just falls and reflects back. In case of transmission, as the name indicates, the electron is going to pass through and the detector is going to be at the bottom. So for electron microscope, it's going to be scanning and transmission. So the right answer is A. For differential interference contrast, we need refractive index, and atomic force usually gives us topographic information. Confocal scanning laser samples usually contain fluorophores, and electron microscopy can be either scanning or transmission. I hope this was clear. So this is a table that states, and you can see on the top panel how the microscopic images look like. So when you compare this TEM and SEM, you can see that the SEM gives you a fair idea about how the surface looks like, whereas in TEM you can see when it is passing through, it's giving a different idea about the same organism. And in confocal, as I told, if you have fluorophores and all, you can see different colors. You can also try to reconstruct 3D images from this. And atomic force, as you can see here, it tells about the hollow places that are present, the highs that are present in it, all of it. I hope the difference is clear, and there was pretty low response in the microscopy questions. So is it clear? I'll just wait for a couple of minutes to ensure that it was clear. If not, please let me know in the chat box so that I'll try to explain. Is it clear? Any doubts? Okay, so in microscopy part, you have to know for what purpose a particular kind of microscope is used, what will be the illumination source in that microscopy, and what kind of images you can obtain from it. So if you know that, you can pretty much link it to the application of that particular microscope. In addition, to be very sure, you can also know up to what resolution the microscopy can work. So this is the basic information that you need to know while you are studying about any microscope. Okay, so the next question is, which organism has an additional layer of periplasmic proteins than which organism from the given options?
Ma'am, option A.
Okay. you can just post your answers in the chat box so that I know that many people are actively listening. I just wait for you to think and answer. Okay, so we'll see the answer for this question, and before that, uh, can you post in the chat box what is periplasm and what is periplasmic proteins? Any answer for what is a periplasm and what are periplasmic proteins? Right. So periplasm is a compartment located between inner cytoplasmic membrane and outer membrane, and this is generally found in gram negative bacteria. So evidently gram negative bacteria has an additional layer of periplasmic proteins than the gram positive bacteria. So if you can see in this slide, we have the difference between what is a gram positive and a gram negative bacteria. So the gram negative bacteria has a complex two membrane system, which uh, we generally call as inner membrane and outer membrane, and in between this there is this periplasmic space. So this contains a high concentration of proteins. Usually 30% of the bacterial cellular protein is going to be in this periplasmic space. So it's going to be the solute binding proteins, transport proteins, chaperones, hydrolytic enzymes. All these are the proteins that are present in the periplasmic space. It is usually produced by the cell and then sent to this periplasmic uh, space. In case of gram positive, they lack an outer membrane. Okay, so they still have a periplasmic space which is kind of a inner wall, and it's termed as inner wall zone. um, so this is going to be between the plasma membrane and the peptidoglycan layer, but Compare to the gram negative bacteria, this is going to be very very small and it's going to be very less complex. So the question was about the periplasmic proteins. So gram negative has additional layer of periplasmic proteins in comparison to the gram positive bacteria. So you can uh, see a gram positive cell has a complex cell wall, peptidoglycan, teichoic acid, all of it is present, whereas in gram negative it looks like a sandwich. And of course on the right side there is this very famous technique through which you can distinguish this bacteria, which is the gram staining technique. So you use different dyes, mordants, wash solutions to identify which kind of a bacteria it is. So first of all you take a sample, add crystal violet, allow it to stay for a minute. So and then you use iodine, which is a mordant. So it makes the crystal violet get attached better to the outer layer of the cell, and then you wash it with ethanol, and finally add safranin, which is the counter stain. Oh, so your gram positive bacteria will look violet in color. So in gram positive the crystal violet stain is going to stay, it's not going to get washed away once you wash it with ethanol, whereas in gram positive your crystal violet is going to be washed away using this ethanol, and finally when you add safranin that is going to stay with the outer membrane, giving you pink color. So with that you can identify which organisms are gram positive and which of these are gram negative. So the other differences is going to be that the peptidoglycan layer is going to be thick in the gram positive organism, and it has teichoic acid, whereas in gram negative you can't find any teichoic acid. So yeah, we of course saw the periplasmic space is absent in gram positive, and the lipopolysaccharide content is very low or none. And based on this membrane arrangement, your flagellar structure will also vary. It has two rings in basal bodies in gram positive, whereas four rings in basal bodies in gram negative. So and depending on this structure only you can find that uh, gram positive organisms usually produce exotoxins, whereas gram negative organisms produce endotoxins. So these are some basic differences that you should know between a gram positive and a gram negative organism. These kind of concepts could also be this is just an extra information that it could also be used in many competitive exams that focus microbiology topic as well. So be it whether it is focused on environmental microbiology or any other microbiology, such concepts are going to be asked in one or two places in any of the competitive exams pertaining to biotechnology or life sciences. Okay, with that we go to the next topic. So here you have to tell which of the following characteristics is metabolically advantageous for a cell. If possible, you can also state the reason. you have heard about a concept called surface area to volume ratio in a cell. So this question was basically focusing on that only. So the surface area of a cell, if I have to explain, uh, is going to be the representation of the cell membrane. Okay. So why we are more focused on cell membrane here? Because cell membrane is the connecting point of the cell to its environment, to release something or to absorb something, to have nutrient uptake, or oxygen uptake, whatever. The role of the cell membrane is very very important. And the surface area of the surface area is the geometric figure that we use while we study this transport through cell membrane. And volume, as we all know, is the uh, internal of the cell, how much volume the cell occupies, like the internal part of the cell has. So the connection between the surface area and volume is very very important. As mentioned, we need shorter diffusion distance for efficient uptake of nutrients, otherwise the cell will start starving. Okay. So in our case, if a cell is increasing in size, it volume grows and the surface area grows, but the rate of volume growth is going to be higher than the surface area. It's very simple mathematics. That how will you calculate volume? By using uh, cube, right? Like r cube. If it is radius and you assume it to be a circular object and you use radius, it's going to be r cube. Whereas in case of surface area, it is going to be r squared. So if the cell size is going to increase, your volume growth is going to be very high, but this should be in par with the surface area. Why? Because uh, when we talk about the surface area to volume ratio, it ideally should be very very high, which means the surface area should be high and the volume should be low. Then only the surface area to volume ratio will be a high value. But why are we more concerned that this ratio is high? Because uh, a high surface area to volume ratio allows high efficient nutrient uptake, because the diffusion distance is going to be very less. Whereas if the surface area to volume ratio is very very low, it means the volume of the cell is very high and the diffusion distance is very long. Therefore it will take more time for the metabolic uptake or the nutrient uptake. So that is why we need high surface area and less volume, so that our surface area to volume ratio is again high, but the diffusion distance is very low. So if this ratio is going to drop, then the cell is going to have metabolic starvation, because it cannot rapidly uptake nutrients from the environment. And also, on the other hand, this waste is going to be accumulated inside the cell. It is not going to be excreted out, because again the distance is long and the travel is long here. Okay. So for metabolic advantage, your surface area to volume ratio should be high, which means you should have a high surface area and a less volume. I hope that was clear. If there are any doubts, again post in the chat box. In the meantime, I have just posted the next question. It's a true or false question, where the start codon binds to the starting sequence of DNA in the protein synthesis. True or false? first slide, which is was nearly the content of this week, had also had this central dogma. So that is why this question was included, and we have different answers. So, what is a central dogma? It is transcription, translation, and replication. So, replication is production of DNA copies, and transcription DNA to RNA, and translation is RNA to proteins in very simple terms. But yeah, the mechanism is very complex. We all have studied all the proteins involved. The mechanism involved in this is really complex, right? We have n number of proteins and other factors that are involved in these processes. So I hope that you are also know what a start codon is. Can someone give in the chat box what is a start codon? Right, it is AUG. And yeah, we also saw in this three domain classification that the first amino acid in protein synthesis is going to be methionine or formyl methionine, whatever, and we have a codon that codes for it. So for whoever who doesn't know what all this means, so in order to synthesize a protein, we need to produce different amino acids. So each codon codes for a amino acid, and a codon is a three triplet. We call it a triplet, like AUG or ATG or whatever. Is that right, whatever I told, or no? Can a codon be AUG as well as ATG? Okay, all of you are silent. Okay, anyways, I'll just keep on explaining. So basically, in any nucleic acid, you can find four nucleotides. So you will have adenine, thymine, guanine, and cytosine. So in case of DNA, you're going to have this ATGC, and in case of RNA, you will have this AUGC. It's not thymine, it's uracil. Okay, and as I told you, there is going to be a specific codon that codes for a particular amino acid, and whenever during this translation process, there is a tRNA that brings this amino acid attached to the anticodon, okay, which is the opposite of the codon that you have. So the statement that is given here is start codon binds to the starting sequence of DNA in protein synthesis. First of all, in protein synthesis, if you are going to consider only translation part, there is no DNA involved here. Okay, we have two processes. So first step is the transcription process. Okay, in that transcription process itself, your DNA is getting converted into a messenger RNA or a mRNA. Okay, and then comes the translation part, where your mRNA already has a start codon in it. Okay, and the corresponding amino acid will now come through the uh, tRNA or the transfer RNA. Okay, so in that case, your start codon does not bind to the starting sequence of DNA in protein synthesis. This statement is false. It's absurd. I hope you got the crux of this question. So if you know the concept clearly, you can just think of where the start codon comes into picture, where the DNA comes into picture, and does both of them have a connection in protein synthesis part? Yeah, they do have a connection in the transcription part, but not in translation, which you evidently call the protein synthesis step. One more question related to this nucleic acid stuff. The small segments of DNA that can move from one region of DNA molecule to another is a what? I hope this is pretty simpler compared to the other questions. Exactly, transposons or jumping genes. Another questions to brush up your basics. Identify the wrong pair. Exactly. So we have two things, purines and pyrimidines. So your adenine and guanine are purines, thymine, cytosine, and uracil. All of these are pyrimidines. So difference is that in purines you have a six-membered ring plus a five-membered ring in terms of the structure, whereas in pyrimidines you just have a six-membered ring. Another question, a couple of questions more, just this and the next one, and with that we are done for the day. You have to identify the false statements. was absolutely right. And Avashri, if I'm pronouncing your name correctly, could you tell why is B also false? Or anyone else, if you can identify it now. Exactly. DNA strands are anti-parallel. They are not parallel. So this statement is false. Okay, so a nucleoside attached to a phosphate group is a nucleotide. That is true. And protein obviously is not a polynucleotide. Maybe it's a polypeptide. And B, two strands of DNA are anti-parallel and connected by hydrogen bonds. If the parallel is going to be replaced by anti-parallel, only then this statement is true. Otherwise, this stands false. And tRNA does not help in the transcription process. We just saw in one of the previous question. So this is the summary of that molecular biology part. So yes, so in the leftmost we have the central dogma where we have replication, transcription and translation. So as I told you, you have a DNA here. This is transcribed into mRNA, tRNA and rRNA. Yeah, obviously they also have to be produced. But in the translation part only, you have the ribosomes. You have this mRNA here, which stands as a template, and you have a tRNA, which brings this amino acid here attached. And corresponding to the code in this mRNA, this amino acid is going to be delivered. They are going to be arranged in the form of chain, and you finally get this protein that is expressed in the cell. So this is the central dogma of biology, molecular biology, or whatever. Central dogma of life, however you want to call it. So in the next, the middle portion, you can see about the uh, nucleotides, nucleic acids, and all. So this image gives the clear difference. If you only have this nitrogenous base, the ribose or the deoxyribose, here based on this, it's called either a DNA or a RNA, and only this part is going to be a nucleoside. Whereas when it is attached to phosphate, it is also called, it is now called as a nucleotide. Okay. And the nitrogenous bases, as we saw in one of the identify the correct pair questions, we have purines and pyrimidines. So adenine and guanine are purines, cytosine, uracil, and thymine are pyrimidines. You can see thymine in DNA and uracil in RNA. And finally, in the rightmost of the slide, you can see the structure of a DNA. The strands are anti-parallel. That's why you have a 5' here, a 3' here on the top, and they are connected by hydrogen bonds. And the different types of RNA are messenger RNA that codes for the protein, the ribosomal RNA that forms the basic structure of the ribosome. And if you rightly remember, we also saw while we were talking about the three domain classification that Carl Woese and his colleagues classified the organisms based on the 16S rRNA, right? So they have some stark variations, they have some stark similarities through which you can identify organisms as well. So that as well you can correlate and remember in terms of rRNA. And finally the tRNA, the coil structure that you see here. So it is basically involved in the protein synthesis as an adapter between mRNA and amino acids. And coming to the final question for the day regarding some transport, the pink is 1 and the blue is 2, and this is a graph between rate of transport versus concentration gradient. Based on this, if one facilitated diffusion or passive diffusion, if one is facilitated, the other would be passive or active. So you can rightly choose. You just have to go through the trend of the rate of transport across against the concentration gradient. Okay, so yes, the answer is A. You can see the explanation here as well. So in case of we have passive diffusion and facilitated diffusion. In case of passive diffusion, yeah, the movement is through the concentration gradient, and it's directly proportional to the concentration gradient, and you can just see a linear line that explains that the movement is linear. Whereas in case of the curve, the red colored curve, it is a facilitated diffusion, because in terms of facilitated diffusion, you have some proteins that is going to facilitate the movement. That is why it is called a facilitated diffusion. Something is going to run the show here. Some proteins are going to run the show. So in that case also, initially your diffusion rate is going to increase only. But once all these proteins are going to get occupied, all the facilitators are going to get occupied, there is not going to be further increase uh, in the rate of transport. It is going to hit a plateau. So that is why uh, the other curve in the red color can be attributed to facilitated diffusion, whereas the blue color curve in the graph is going to be attributed to passive diffusion. In case of active diffusion, it's going to happen against the concentration gradient only. hope it was clear. So, with this, we conclude the session today. So, before you leave, I would be very grateful if you can post in the chat box whether the session was useful or whether you need some modifications in the session. As I informed prior, before starting with these questions and all, I prepared this based on the understanding of how I handled sessions in the previous for the previous NPTEL courses or for the same course a couple of years back as well. Uh, and I was very happy to know that many of you are from the life sciences background. So maybe these questions seemingly were very basic to you. If that's the case, you please post in the chat box, or if you want me to host the session in some other format, that also you post in the chat box so that I'll prepare for the next session accordingly. If not, thank you for joining for the session today. Hope to see you all in the next session as well. And really thank you for being so active, because my session was full of questions, and without your participation it couldn't have been uh, so good to host. Thank you so much.
Thank you, ma'am.
Thank you. If you have any doubts or feedback, please feel free to share it in the comments, or please unmute yourself and you can just tell me. Any more doubts or comments, or shall we close the session?